So welcome also on my behalf to this webinar, and greetings from some sunny Finland. Very nice, warm spring weather here, but for reasons we all know, very few people outside and most of us sitting inside and working from home. So my name is Nina Aro. I work at VTT, Technical Research Center Finland, as a principal scientist and project manager. I have been working at VTT in the protein production team for over 15 years. In our team, we develop protein production systems in microbes. We focus on understanding the molecular background and physiology of protein production, and learnings from these studies are then used for improvement of our production host. So at VTT, we have over 2,000 employees, and our focus as a research organization is to solve the current global challenges, such as resource sufficiency, climate change, and sustainability. In our protein production team, we try to tackle these challenges with the help of proteins. So, for example, for the renewable biofuels, we work on discovering novel enzymes and novel enzyme activities, and engineer production hosts to produce single enzymes or enzyme mixtures for biomass hydrolysis. In the field of renewable materials, we have, for example, engineered our host to produce structural proteins such as silk and elastin, and combined these with nanocellulose to produce new composite materials with interesting properties. In the pharma field, we have generated production hosts producing, for example, antibodies and vaccines or more affordable therapeutics. And maybe the newest field is relates to protein production for food supplies. So we have developed technologies to produce animal proteins, such as milk whey proteins and chicken egg proteins in microbes, without so diminishing the need for animal production. So this slide lists the microbial hosts we have worked with. So we have some work done with E. coli, and then quite a lot of work done with yeast hosts, Saccharomyces, Pichia, and Cluvaromyces. Pichia here, the most common one. Then we have developed expression hosts of filamentous fungi, Trichoderma reesii here, the most used one, but also a lot of work done with Myselioptera thermophila and some work with 2 different Aspergillus species listed here. Then we also have a separate team working with plant and plant cell cultures. Then as an example for proteins that we have produced, these are now examples that are from our own internal, mainly from our own internal projects. We have, of course, quite a lot of numbers over the years from our customer projects, but those I am not able to share here. But as you can see, the first 3 ones are Trichoderma enzyme or fungal enzymes produced in Trichoderma. Here the production levels range from 8 to 28 grams per liter. Then we have 2 examples from the pharma side full-length antibody, 7 grams per liter, and antibody Fab fragments, 8.2 grams per liter. Then a structural protein, spider silk fusion protein, produced at 4 grams per liter. Chicken of albumin, produced at 4 grams per liter. And then a fairly recent result from Myselioptera thermophila. This is done with a collaboration with a customer. That's 24.5 grams per liter of a full-length antibody. A vaccine, 6 grams per liter, and xylanase in Pichia pastoris, produced at 6 grams per liter. A lot of these numbers are from the first generation strain without very much product-specific optimization. So, for example, for the xylanase in Pichia pastoris, that is the expression level that we get with our sort of standard production procedure. So regarding large scale, when we think about large scale production, we really think about productivity. How many grams per liter of our protein of interest can we produce? How many grams per liter in an hour? And how many grams per gram of substrate. So I have listed in the next slide some key features of our 2 most used production hosts, Trichoderma ricii and Pichia pastoris. Trichoderma is a well-known industrial producer of cellulotic and hemicellulosic enzymes. It's been used very long time for production of enzymes for food, feed, and textile application. It's a very efficient protein secretor. The highest result at VTT is 110 grams protein per liter. Trichoderma has a GRAS status, so it can be used for manufacture of food proteins. For Pichia pastoris, it's also a well-known industrial production host for many non-pharma and pharma proteins. It is easily genetically manipulatable, fairly fast to manipulate, and can be cultivated in inexpensive media. It is also a very efficient protein secretor and has GRAS status. So when we think about large-scale production, we really have done quite a lot of work to optimize our production strains to obtain higher yields, constantly higher yields. So in the next 2 slides, I have gathered 4 examples of these improvement strategies that we have done. The picture on the left-hand side illustrates total protein production levels in base strain, marked with blue circles, and then an improvement of the strain by overexpression of a transcriptional regulator, giving about 20 grams per liter improvement in protein production level. This is data is from a fermentation with optimized bioprocess using cellulose as the main carbon source. The slide next to it, or the picture next to it, shows then the work we have done to reduce protease activity in our strain. So Trichoderma is a host that in the same conditions where it produces a lot of proteins, it also produces a lot of proteases. And this is a problem if your target protein is very susceptible to proteases. So in the right-hand side, the bar on the right-hand side illustrates the protease activity of in our base strain, given the value number 1. And then we have identified the most common proteases secreted into the culture medium and deleted them one by one, leading to reduction of the protease activity found in the culture supernatant. Deletion of 7 proteases has reduced the original activity to 4%. We have our current strain is deleted for 11 proteases, so quite many of our target proteins are very stable in the culture supernatant of this strain. But sometimes it is needed to go beyond that, and for that we have identified more proteases that can be deleted or protease regulators that can be modified to reduce the protease activity in the strain. The next slide shows then development of mutant strains that allow production of glucose. So in the previous slide, I showed the production on cellulose, but cellulose as a solid material is not, in some cases, is not a suitable carbon source for large-scale production, but instead a soluble carbon source is needed. So for this purpose, we have developed a mutant strain where cellulase genes are not repressed by glucose, but instead are activated. So this figure on the left-hand side shows again our base strain total protein production level in gray circles during cultivation of glucose. And then, so as you can see, that produces less than 10 grams per liter total protein. And then our modified strain expressing a mutated transcription factor producing about 60 grams per liter total protein in glucose cultivation. Then in the figure on the right-hand side of this slide shows the development of synthetic expression system that we have developed at VTT. This system consists of 2 parts. The expression cassette for a synthetic transcription factor. Synthetic transcription factor consists of 2 parts, a DNA binding domain and a very strong activator domain. And then the second part of the system is the expression cassette for your protein of interest. In this cassette, the expression of your protein of interest is controlled by a core promoter that is activated by the synthetic transcription factors binding in front of the core promoter. The expression level can be tuned by varying the number of binding sites for the synthetic transcription factor. But usually when we are thinking about large-scale production of single enzyme or single protein, we really go with the maximum number of binding sites and aim for the highest production level. In the STS page picture below, I try to illustrate the comparison between the synthetic expression system marked here CES, and then our traditional CBH1 promoter-based system marked here PS. So when cultivated on inducing media, spent grain lactose here, you can see that the expression of our protein of interest, in this case CPH1. So in the CES strain, the CPH1 gene is controlled by the synthetic expression system or the synthetic promoter, and in the PS, the CPH1 gene is under its own promoter. So when we cultivate on inducing conditions, you can see the CPH1 band here and quite a lot of other proteins being secreted. But this here you can see that the expression level from the CES promoter and the CPH promoter is very similar. But then when you compare the same strains in a glucose cultivation, you can see a difference that the CPH1 promoter is repressed in the presence of glucose, so you don't see practically any CPH1 in the culture supernatants, whereas using the CES promoter and the CES system, you can see very clearly the CPH1 band in the culture supernatant, and actually then no other or very few other proteins being secreted as they are repressed by the presence of glucose. This is the CES system is especially nice when you are aiming to produce one protein or just one or two proteins in one strain. And the nice advantage about using glucose is that it is readily available all over the world. It's fairly cheap and enables a relatively simple production process. I have here then 3 example cases for projects that we have carried out in our research team, taking advantage of the improved strains that I described in the previous slides. First example case is about screening of novel xylanases using the CES system. So VTT has a culture collection of its own, and we have carried out a project to search for xylanases with improved properties. We took several fungi from the culture collection, cultivated them on media that contains plant polysaccharides such as cellulose, xylane, pectin, for induction of polysaccharides encoding enzymes. The mycelia was collected and RNA was isolated from several time points and from several different culture conditions. The RNA was pooled and sequenced and assembled into VTT in-house database. From this database, we selected a set of xylanases to be expressed in Trichoderma ricei. These genes were cloned in the CES vector under the CES promoter, and the constructs were transformed as a pool to Trichoderma, and then transformants were analyzed for xylanase production and activity for xylanase activity. As you can see from the STS page shell in this figure, we get quite nice production of xylanases with very low background and also different levels of production. The advantage of being able to screen in Trichoderma, especially if you are planning on constructing, or if you're planning that your final production host is Trichoderma, is that already in the screening phase you get an idea that how well is the gene or how well is the protein produced in Trichoderma. The strains that come out from this screening process are definitely not the final production strains. but you get an idea whether your enzyme is easily producible or not. The second example is about production of cellulase mixtures for biomass hydrolysis. So we have base strains that produce quite nice total cellulase mixtures, and usually we initiate a project like this that we identify the bottlenecking enzymes for hydrolysis of that particular biomass of interest. We screen for the suitable enzyme candidates to improve the enzyme mixture produced by the base strain. Then we transform the desired enzyme genes to our trichoderma strains. We have several different promoters available, leading to different production levels, and several loci identified for strong and stable expression. We can transform one expression cassette at a time, and after each step analyze the limiting enzyme activities, or we can transform several cassettes simultaneously using CRISPR transformation. And then once you have the final strain, then of course you do development of production process, and we can test different raw materials for carbon and nitrogen source. On the right-hand side, I have two pictures showing the development of enzyme mixture. The upper picture shows biomass hydrolysis of pre-treated softwood biomass. So the enzyme mixture produced by our base strain was releasing about 55% of the glucose of theoretical maximum. Then when we added the first enzyme, in this case it was beta-glucosidase, to our strain, or overexpressed beta-glucosidase in our strain, the released glucose level was already increased to 80% of the theoretical maximum. Then a second enzyme was added to the strain, that was cellobiohydrolase, and again the hydrolysis of the enzyme mixture was improved. And then the third, after the addition of the third enzyme to this strain, we are now at a level where the enzyme mixture is able to release 94% of the glucose of the theoretical maximum. Another, the picture below shows another enzyme strain development work where we have generated 3 different strains marked here as SF4, 5, and 6, and produced enzyme mixtures by these strains and compared the enzyme mixture performance to Celic Seedtech 2 in the hydrolysis of wheat straw. And it's a little bit boring picture since the curves are very much on top of each other, but showing very nicely that with relatively simple overexpression of selected genes, we can generate fairly good strains producing fairly good enzyme mixtures. These strains can then be further developed depending on the biomass and the pre-treatment used for the particular biomass. The third example is production of proteins in bioreactor using Pichia pastoris as a host. Here we have 3 proteins. LGB is the cow milk beta-lactoglobulin. is a bacterial phytase, and then we have a bacterial xylanase. Here we have used the CES system for production, and as you can see in our Pichia host, all of these three proteins seem to be very stable. There's not very much degradation, no degradation seen, and the production level ranges from 1.5 grams of LGB being produced to up to 6 grams per liter xylanase. These are now with the sort of standard procedure and can be or will be optimized further for each protein of interest here. This slide summarizes the upscaling facilities we have available at VTT. So we usually do the first screening cultivation and small-scale cultivations either in a 24-well plate format, in a normal shaker, or we use the biolector, where you have the 48-well plate cultivations, and already quite a few parameters can be controlled and we can monitor the growth. Then the next scale is amber, where we have 24 parallel bioreactors and 250 mil cultivations. Here you can control pH, aeration, oxygen, control the foaming by anti-foam, and automate the sampling. In the biolector scale, trichoderma generally produce 2 to 6 grams per liter, and then in the amber, the production level can be up to 50 grams per liter, depending on the protein and the strain. And then the next step from amber, we have quite a few reactors ranging from small scale to intermediate scale, 1 to 30 liters. And then we have pilot scale reactors from 300 to 1200 liters. In this scale, the production can go up to 100 grams per liter, like I showed in my previous slide. Trichoderma in general is fairly well scalable from 1 liter to 1,000 liters. But of course, work needs to be done there also. Then we have downstream processes, equipment, for example, drum filters, continuous separators, and equipment for ultrafiltration. On the right-hand side, I have few examples of enzymes that have been produced at large scale at VTT. So several grams of CBH1, 8 grams of LPMO cell 61 has been produced, and 10 grams per liter of neurospora beta-glucosidase. These production levels enable quite many application tests to be done in an industrial scale or scale close to industrial scale. Our bioprocess theme, the research interests are, of course, optimization of cultivations and production conditions. They they carry out physiological studies and characterize the strains we have generated in the protein production team for us or for them. They have quite a lot of examples on bioethanol, beer, xylitol production, and of course enzymes, as I mentioned before. The bioprocess optimization involves the development of feeding strategies, development of continuous cultivations, of course, issues related to scale up and related to bioreactor development for a specific process, and development of downstream process for protein of interest. And as a last slide, I have here that we also at VTT provide sustainability assessments for different production processes. These might be important to carry out at a specific phase of your production strain development, and it might help to decide what is the kind of process you want to develop further. So this involved the techno-economical assessments, concept specification, mass and energy balance, process simulations, analysis of costs, CapEx and OpEx, and sensitivity analysis. Also, we do environmental assessments, LCA analysis involving carbon footprint, water footprint, and then other modeling approaches. So a nice full package of information available. I think this was my last slide. Thank you very much for listening.